Good morning, dear participants. Am I audible?
Yes, sir. Ganesh, sir. Yes.
Yes, sir. Yes. Good morning, everyone. Welcome to the day 5 of 1 week national level online training program on network pharmacology. I believe the last 4 days are very fruitful, engaging, and interesting to all the participants. One more engaging and interesting session is waiting for you. Today we have with us Dr. A.K. Krishnaswarup, young and dynamic faculty from Sandeep University. I personally believe he is going to drive you deep into the molecular modeling tools, which are very much essential in integrating network pharmacology. And I request all the participants to utilize this opportunity and make use of this resource person who is very well versed with all the softwares. And he has done a lot of research work, especially in molecular modeling as well as network pharmacology. With this, I would like to invite Dr. Sumit Devre, sir, Professor, Department of Pharmacognosy, School of Pharmaceutical Sciences, Sandeep University, to introduce today's resource person.
Good morning, one and all. I am Dr. Sumit Devre. As our coordinator, Dr. Gandhi sir told regarding Dr. Swarup. So I am here to give his brief introduction. So Dr. Akai Swarup is accomplished researcher in field of pharmaceutical chemistry. He has earned his bachelor degree from Sri Vishnu College of Pharmacy and his master's degree from JSS Academy of Higher Education, Mysuru in 2021. In his PG work, he has received prestigious award, that is PG Pharmacy Fellowship, Rangachari Memorial Scholarship, and also from Tata Education Development Trust. His major work, it is on modulation of pro-inflammatory molecules. Sir has did his doctoral work under Dr. S. Jubi Madam from JSS Academy. He received travel grant as well as a research grant from JSS Academy. Regarding research, his productive research, he has authored 29 publications with cumulative impact factor of 63.7. Sir has contributed 3 book chapters, 2 newsletters, and hold 1 grand patent, and many more are yet to published. His acknowledgements or his achievements, he has received 6 best poster, oral and paper awards. He got best outgoing researcher award from EPTI and best research scholar award from JSS College of Pharmacy. With this brief introduction, I introduce Dr. Akai sir. As sir, he is working with Sandeep University, Department of Pharmaceutical Sciences. Thank you.
Thank you, sir, for your brief introduction. Sir, can I start, sir?
Yeah,
One second, sir.
sir, you can start.
Sir, you are host now. You can start the session.
Yeah, my screen is visible, sir. My slides.
Yes, sir. Yes, sir. It's clear.
Yeah. First of all, I want to thank you, Dr. Sumit, sir, for giving a brief introduction about me and my research work. And I also want to thank our today's coordinators, Dr. Mohan Gandhi sir and Nagaraju sir from the School of Pharmaceutical Sciences, Sandeep University, for giving this opportunity. And myself, Dr. A.K. Krishna Swarup, Assistant Professor from Department of Pharmaceutical Chemistry, School of Pharmaceutical Sciences, Sandeep University, Nashik. Today, here I am going to discuss my topic on integrating network pharmacology and molecular modeling tools in pharmaceutical research. As we have seen this training program from the last 4 days about network pharmacology, various aspects and various approaches, various benefits. So I don't want to drag about the introduction much, and I just want to give the glimpses of network pharmacology approaches and benefits, and some case study regarding the project work what we have done in network pharmacology. So coming to network pharmacology approaches, as we all know, the traditional approaches before coming network pharmacology into the field, it's like one target, one drug, one disease approach in the drug discovery. So all we have take only one drug and one target and one disease. So simple, we are going with the simple target by taking small drug, and we are going for the efficacy, safety, and sustainable challenges. And there are some limitations like safety issues, efficacy issues, and sustainable issues. Now, a new discipline called network pharmacology has been emerged, which attempts the drug action and interaction with multiple targets. So, what here we are doing in the new emerging field network pharmacology, as traditionally we are taking only one drug, one target, and one disease. But in new emerging field network pharmacology, we can see the multiple target of action in a disease, because this disease is a complex pathogenesis. It consists of multiple molecular pathways. So we can approach by using this network pharmacology, we can identify which targets are majorly responsible for the disease progression, or which type of drugs are helpful for this multiple target for disease progression. So how we can use this network pharmacology? How we can use by using some computational power. As network pharmacology approach is one of the computer-aided drug design approach as from for the drug discovery. So it is systematically catalog of molecular interaction of drug molecule in a living cell. In a living cell, what type of molecular interactions are happening? We can computerizely visual that interactions, and we can say, okay, this drug is acting in this specific pathway. This disease is okay. Like that, we can see from the network pharmacology approach. And it also attempts what is this major application is new drug leads and targets. We can discover a new drug lead, and also we can go for the repurposing of existing drug molecules. Already, for example, if a drug is having an anti-cancer activity, maybe the same drug will have the activity on anti-inflammatory or anti-neuro, any Parkinson or depression. Maybe we can identify through this network pharmacology approaches. A distinctive new approach to drug discovery can enable to paradigm shift from high specific magic bullet based drug discovery to multi-targeted drug discovery. As already we said, from the updated version of research, bullet based drug discovery means only one drug, one target to multi-targeted. We came into the updated version in drug discovery, which is multi-targeted drug discovery. So ultimate our goal of network pharmacology is to get the drug with safety, efficacy, and very good action in the disease progression. So this is the scientist Hopkins who proposed the network pharmacology in 2007. As we are seeing this network pharmacology emerging into the drug discovery processes. So there are some optimization techniques from network pharmacology, like identifying drug interactions, like mapping drug interaction with multiple targets and discovering the new targets, finding new potential target from the drugs, finally repurposing of existing drugs. So ultimately, our aim is to get a lead molecule. We want to filter and identify the lead molecule for the particular disease and particular target. So this network pharmacology is very much useful for these approaches. So, what are the benefits of this network pharmacology? As already I said, comprehensive analysis. We can know the drug effects and off-target interactions and rationally drug discovery. For many researchers, they can identify the various targets in the disease and identifying the novel targets, repurposing opportunities like offers cost-effective drug development strategies and data-driven prioritization. For these are the major benefits for the network pharmacology approaches. Coming to network pharmacology in research aspects. So everyone wants to start the research work. At the beginning, we'll think what research we want to do, what studies we need to carry out, what phases we need to develop, what are the objectives we need to keep, what are the final output. So if you want to start as a network pharmacology as a base from the scratch to preclinical studies, we can do the project works or research works in the network pharmacology approaches. So in the network pharmacology approaches, as we selected any disease and we can find out the targets and what are the compounds. As we today we are seeing about the traditional medicines, like modernized traditional medicines like plant constituent, plant metabolites, what are the compounds having which type of activity, which type of proteins we need to select. By using network pharmacology, we can generate the data and we can create the protein interaction and protein drug interaction networking and fun enrichment analysis like gene ontology, cellular ontology, and different pathway makers like KET pathway, reactome pathway. We can identify the pathway and finally identifying the lead compounds and lead target from the disease. Then we go for the molecular docking studies and further we can go for dynamic studies also. And finally, which compounds are acting as the lead, we will go for the in vitro studies like cell viability by MTT and whatever the studies by using the protein inhibition by ELISA, whatever it may be. And finally, in vivo animal studies. This is a preclinical study. From the starting from the scratch, taking the disease and identifying the target, taking the plant or plant constituent, and finally optimizing the uh, drugs, whatever whichever drugs are possible to inhibit the target. So we can identify by using these techniques. This will be helpful for the research works. So network ethnopharmacology, as today's FDB like e-training program says about the natural products like traditional medicine, herbal products are useful for the research. So during the progression period of network biology, natural products were gaining importance in the chemical space of drug discovery because natural products has been designed and synthesized by the nature for the benefit of evaluation. Whatever we are taking from the nature, it is an evaluation for the benefit of any disease or any particular use of that natural constituent. And development of many databases from the botanical information and powerful tool that bridges the traditional medicines and modern molecular biology. And the network-based approaches provide a systematic platform for the study of multi-component traditional medicine and applications for its beneficial modernization. So these natural compounds are very helpful for the modernization pathways and what modernization molecular biology tools. So these are some of the introduction about the network pharmacology, pharmacology regarding. Now I will start with a case study, which is very interesting. So for example, this is the case study what we have done in our lab. So uncovering the potential active constituents of Parisia americana for rheumatoid arthritis treatment using network pharmacology. This is our case study. The aim of our project is to identify potential therapeutic targets and natural compounds for the treatment of rheumatoid arthritis, a chronic autoimmune disorder that affects millions of people worldwide. Already we know that rheumatoid arthritis is a chronic autoimmune disorder. It affects the millions of the people in the worldwide. So as we know, rheumatoid arthritis is a complex disorder, which signaling pathway is major important, which targets are major important. We can't know as starting of the disease. So based upon the literature, we can find some targets based upon some research was already conducted. We can know the targets. So in this study, we using network pharmacology to identify the target, which potential target is used for the rheumatoid arthritis. To achieve this, we use the bioinformatics tools to collect and analyze the data on the phytoconstituents of Parisia americana, which is nothing but avocado, and identify the common genes between avocado and rheumatoid arthritis. So as I said about the rheumatoid arthritis, which is a chronic autoimmune diseases characterized by the inflammation and joint destruction, leading to significant disability and decreased quality of life. So where it will be happens in rheumatoid arthritis, commonly affects the joints in the hands, wrists, knees, and joint lining becomes inflamed, causing disease of the joint tissue. And rheumatoid arthritis major symptoms are bruised, heated, and swollen joints, usually worse in the mornings, joint stiffness, fatigue, fever, and loss of appetite. These are some of the symptoms of the rheumatoid arthritis. And coming to Persia americana. So for this, we know as rheumatoid arthritis is an inflammatory disorder. For the selection of the plant, so which type of plant or which type of fruit we need to select as a traditional medicine for the inflammatory purpose. So we see that avocado having some traditional use of anti-inflammatory from the literature search, and we have selected this fruit is our choice of plant, and in which this avocado, which constituent is major responsible to decrease the inflammation in rheumatoid arthritis. So avocado has a long history of traditional knowledge, which serves as a foundation for exploring its therapeutic potential in context of rheumatoid arthritis. And there are many bioactive compounds like including polyphenols, flavonoids, and fatty acids. These compounds have shown anti-inflammatory and immunomodulatory properties. And coming to the safety and availability, as avocado is generally considered safe for consumption, safety is crucial when exploring potential treatments for chronic conditions like rheumatoid arthritis. So easily available compound in the market, avocado, and safety for consumption. Many bioactive constituents are present in this. So we will identify which constituent is helpful to reduce this inflammation in rheumatoid arthritis. So we will take the constituent and through network pharmacology. And this research gap and hypothesis, why we need to do this rheumatoid arthritis and why we need to select this avocado. So this is the disease prevalence. So already we've seen the rheumatoid arthritis is a highly prevalence and substantial impact on patient quality of life, make it a significant health concern. Their unmet medical needs. Like although there are treatment options are available for rheumatoid arthritis, like NSAIDs, DMARDs, disease modifying associated with rheumatoid disease, the drugs, there is still an unmet need for the more effective and safer therapies. So from the natural therapies, it will be the best suit for the rheumatoid arthritis treatment. Many Ayurvedic formulations are also available. So maybe avocado also one of the major traditional plant which is also helpful for the rheumatoid arthritis as we have thought and we started this work. And rheumatoid arthritis is a complex pathogenesis, like studying the underlying mechanism and potential therapeutic targets in rheumatoid arthritis can provide a valuable insights into the disease pathogenesis and aid in developing targeted interventions. So this is our gap, and we are going for the study. Our project seeks to fill this gap and contribute to the scientific understanding of avocado potential as therapeutic option of rheumatoid arthritis. So as I said, rheumatoid arthritis is a complex pathogenesis pathways. So these are the many signaling pathways are involved in the inflammation of rheumatoid arthritis. Like many pathways, signaling pathways, somewhere the rheumatoid arthritis is increasing by this one of the pathway. So from the literature search, I am finding some pathway is important, some pathway is important. So we have a little bit of confusion how to select the which pathway. Through network pharmacology, we can identify the best pathway regarding to the avocado constituents. So here many inflammatory pathways are there, like the JAK-STAT pathway, like one of the pro-inflammatory cytokine like interleukin 6 or 17, interleukin 2. These are the major pathways which will activate the JAK kinase pathway. It will activate the STAT, finally undergoes the inflammation. So these are many signaling pathways are available in the rheumatoid arthritis. So which pathway we need to select, which constituent we need to take through network pharmacology, we can explore more. So tools used in this study. So today's I am explaining about these tools, various tools for this study. Like already from the last past 4 days, we are seeing some of the tools are repeatable, but today I will explain some more deeper. Like impact database, like Indian medicinal phytoconstituents and therapeutics, which is a database where it contains the phytoconstituents of almost 17,000 phytoconstituents are available from this database. And coming to the Open Bible, this is a software like which we can convert which type of format. Like there are several formats in the drugs like SDF format, MDL format, PDB format. This software will helps to conversion of one type of format to another type of format. For example, some softwares will accept only the PDB format. I have SDF format. This Open Bible software will help to conversion of this SDF format into PDB format. So the software helps to conversion of the formats. And coming to the data various software. This data various software today we are using to check the ADMET properties like absorption, distribution, metabolism, toxicity studies. By using this data various software, we are predicting the ADMET properties. Next, coming to the binding DB. So for this, we are collecting some of the genes and binding targets. For that, binding DB web server is used. And next, DisGenet. This software is used for majorly identifying the disease-related targets. Disease-related targets are identified by the DisGenet, and binding DB is used for the identifying the targets from the phytoconstituent or any drug molecule targets. And next one is Veni. Veni is used for the identifying the common targets which are available from the phytoconstituents and disease targets. And coming to cytoscape, here the cytoscape plays a major role to create a construction of network where we can construct protein-protein interaction networking and protein-drug interaction networking from the cytoscape software. And next, some molecular modeling tool like Pyrex, which is an molecular docking software, and Storage Studio Visualizer. These are the some softwares today which I was going to explain related to our project. So for every work, there will be a plan of work like literature survey, target prediction, common genes, analysis of protein-protein interaction and protein-drug interaction, and molecular docking. So through literature survey, what we can gather about the disease, rheumatoid arthritis diseases, and about the Persona americana, which is avocado. What are the phytoconstituents? Till now, what are the research has happened? Which type of phytoconstituents is helpful? And rheumatoid arthritis, which signaling pathways is majorly helpful? This type of information, till now, what are the drugs available? This type of information we will gather from the literature survey. The next one is target prediction. Using binding DB bioinformatics tools to predict the targets for the selected phytoconstituents and DigiNet to identify disease-associated genes for the rheumatoid arthritis. So we'll start with the target prediction. Yes, the first step in the left-hand side, we have taken the Persea americana, nothing but avocado. And in the right side, we have taken the rheumatoid arthritis is our disease. So it is a flowchart. We can clearly see which steps first I'm going to do. So Persea americana, going to the impact database, which is nothing but phytoconstituents what we collected. So in the first step, we are collecting the phytoconstituents regarding all the avocado constituents. Whatever avocado constituents are present in this database, all the constituents we are collecting through this impact database. After collecting these constituents, so there are many number of constituents means we need to go for the filtration by using ADMET screening. So based upon the ADMET screening, we are going to filter some of the constituents. By using data various software, we are screening these constituents. After screening, whatever the constituents are present, for that we are identifying the targets. For the each constituent, we will identify the targets by using binding DB software. So with the help of Uniport, exactly what type of gene is present, we will identify and targets of Persea americana we will collect in one side. All the targets of avocado we will collect on the one side. In the right hand side, rheumatoid arthritis, we will go for the DisGenet web server. In this web server, disease associated genes. There are another more servers are there like gene cards, mala cards, whatever the we can go for different web servers also. So right now we went into the DisGenet web server. So from this we collected the targets of rheumatoid arthritis. So one side we collected what we collected targets of Persea americana, which is nothing but avocado targets. One side we collected the rheumatoid arthritis targets. From this we are going to for the network pharmacology. So each and every step now I will show what we have collected. As I said, first step is IMPACT, which is Indian Medicinal Plants Phytochemistry and Therapeutics. From this database, from the avocado, we have collected total 253 phytocompounds. Total 253, whatever we have collected these phytocompounds. Out of these 253 compounds, 98 were fruit and 155 were leaf constituents. So all the 253 in that 98 were fruit and 155 were leaf. We collected and we have downloaded each and every compound we have downloaded and we are prepared this molecules by Marvin's case software. followed by. So this one we are want to go for the ADMET properties. For this, we need to convert these molecules into mole SD to mole SDF format in the open Bible software. So I will show the software how it will be works. So normally this is the software of open Bible. Here you can input what file you have, like mole, SDF file, or in which type of molecule it is there. Mole or mole 2, SDF, PDF, PDB, PDBQT, whatever the type of molecule is there, you can input and output. You can clearly see in here which type you want. For example, I have the compound of MDL mole format. I want to convert into PDB molecule. So you can input the molecule, and by using the convert option, you can convert the molecule. So like this, by using the Open Bible software, you can convert what type of format into which type you need the format, we can convert. So like that, we converted for all the compounds. So this is the where the impact, where we can identify the plant constituents. So after collecting all the 253 constituents, the ADMET analysis were carried out by using data various software. Uh, for checking the Lipinski rule like molecular weight, log P, log S solubility, partition coefficient, hydrogen bond donors, acceptor, rotatable bonds, toxicity assessment like mutagenicity, tumorigenicity, reproductive effect, and irritant. These are the properties where the molecule should have within the limits. If the limits are not in, if the constituents are not in the limits, there will be some problem in like bioavailability issues and whatever the things toxicity issues may come. So for the screening at the beginning only, we are conducting the ADMET screening. From this, number of constituents will be filtered so that we can go for the safe and efficacy drug molecule throughout the study. Based upon these criteria, out of 253 constituents, 64 active constituents were selected. In that, 17 are from the food and 47 from the leaf. So almost we reduced it to 64 active constituents. So this is the software where data various software. So many people can use like Swiss ADME, Protox, several ADME web servers are there. But why I have utilized this data various software? So here you can utilize all number of compounds at a time you can input into the software. How many, for example, 64 constituents or 250 constituents, we can input all the compounds at a time into the software. So in the right side, you can visible total molecular weight, partition coefficient, solubility, hydrogen bond donors, acceptors, donor. You can directly filter here itself directly. For example, molecular weight, I should have 100 to 500. I will keep the limits here only like directly by moving this option. In log P, partition coefficient, how much I need, solubility, how much I need, hydrogen bond accept, how much I need. So based upon that, all the constituents we can filter within a minute, and we can go for the next step. So by this software, I used this type of ADMET analysis. So if you use the Swiss ADME also, the same result will come. Protox also same result will come. Already we have tried. So for shortening the work, we have used this software. So these are some of the ADMET properties of the food constituents, top 10 food constituents, like molecular weight should be less than 500. And what is the criteria we have? Partition coefficient from -2 to +5, and solubility, hydrogen bond acceptors 1 to 10, hydrogen bond donors 1 to 5, polar surface area less than 140 angstroms, rotatable bonds, and some toxicity like mutagenicity, tumorigenicity, and irritant effective. These are the should show all the things within the limits. Then that compounds only we selected for our further studies. So these are the ADMET properties of the food constituents, and these are the ADMET properties of the leaf constituents. I have kept top 10 compounds. So after completing of the ADMET properties, we got 64 constituents. Next, we are identifying the target prediction. How we will identify the target prediction? I already said from by using the binding TB database, we are going to the identify the targets. So the same thing from the impact database, you can collect the SMILES notation of all the 64 compounds, and you can paste in the database binding DB software. Here, based upon that, you can identify the targets. So this is the binding DB web server, which how it will visualize. Here you can go for the special tools. There you can find the targets. So you need to enter the smiles. Then directly you can upload which constituent you need based upon the similarity score. You can identify the targets. So for all the 64 constituents, we need to identify each constituent targets. So, but for me, based upon the similarity score 0.5, which was considered, we got only for 25 constituents only we can gather the targets from the binding DB software. So out of these 25 constituents, 4 are from the fruit and remaining 21 from the leaf. So this total number of targets we are getting from the 25 constituents are 499 genes. But in these 499 genes also many genes are repeatable. So we have filtered again these 499 genes. So finally we obtained 209 genes from the Persea americana. So as from the starting from 253 genes, 253 constituents we have identified. From the ADMET we filtered into 64 constituents. From that 64, we are identified the targets of 25 constituents. From that, we have identified 209 genes. We obtained total 209 genes from Persea americana. So this information was then used for the perform protein-protein interaction networking analysis and identifying the top genes for further analysis. So these 209 genes we are carried out for the next step. So next step is, as one side we have collected the Persea americana targets. In the right hand side we have the rheumatoid arthritis. So we need to collect the rheumatoid arthritis targets also. As DisGenet's web server, you can directly Google in the DisGenet's web server. Directly you can pick the rheumatoid arthritis. So you can select the disease associated genes. There are total 2723 target genes of rheumatoid arthritis is there. So that and all we have downloaded the genes. So the next step is Veni. For what reason this Veni we are using for this network pharmacology. So as I said, 209 genes we have isolated taken from the Persea americana and 2723 from the rheumatoid arthritis disease protein targets. From this we can identify the common targets. What are the common targets in the Persea americana? What are the common targets in the rheumatoid arthritis? So these will helpful for the disease progression. These are the major for the Persea americana also. These are the targets which are important for rheumatoid arthritis also. These are the targets important. So from this we got 86 targets which are common targets. So these are the Venn diagram. You can see in the left hand side rheumatoid arthritis total 2637 plus 86 targets and Persea americana 123 plus 86 which is 209. So these are the 86 common targets we have taken. So for your better understanding, I will show this one easy way. So already I have said the constituents which I have collected. So these are the rheumatoid arthritis genes which I have collected, and these are the different from fruit constituents and leaf constituents we have selected. So you can take that rheumatoid arthritis genes. So after you can go for any point. Easy way of sorting of common genes. So in the list one, you can keep the rheumatoid arthritis like RA. In the left hand side, you can keep the Persea americana, which is nothing but avocado. Here you can copy the all the targets which are taken from the arthritis. So here you need to take the constituents from the Persea americana. So based upon this, we can identify the target genes easily. So one side you keep the target genes. So you can clearly see what I showed the result, which is total 2637 genes and Persea americana is 209. So 123 plus 86 is nothing but 209 genes. So these are the common genes. So how we need to get these common genes? So if you click on the 86, automatically you will visualize the 86 common genes which are produced. In this way, we are identified the common genes from the Persia americana and rheumatoid arthritis. So we have identified the target identification and common genes we have identification. Next, we are going for the doing the protein-protein interaction networking and protein drug interaction networking by using cytoscape software. So in order to analyze the protein interaction, 86 drug disease interaction genes target were analyzed using PPI network affected by the string database. From this string database, we have developed the protein-protein interaction. We have got 86 nodes and 527 edges, indicating the complex network of interaction between targets of Persia americana and rheumatoid arthritis. As already I have said, what to do this 86 targets. We are conducting the protein-protein interaction by using the cytoscape software. So this is the software of Cytoscape, how it will be visualized. So here we need to incorporate that 86 target genes, and we need to analyze the protein-protein interaction by using some plugins. In this software, you will have different plugins will be there, like CytoHubba, CytoMCA, Strings, different options we will available. Based upon the CytoMCA, you can identify which targets are important based upon the degree betweenness closeness. So already from the previous lectures, you can understand what is degree, what is betweenness, what is closeness. So from this, you can see, for example, this PTGS2 gene is there. This is called whatever the circles you are seeing, which is nothing but nodes. Whatever the circles, what you are seeing is nodes. What are the lines you are seeing, each target to each target is called edges. Based upon this only, the degree betweenness closeness will be calculated. So, for example, here how we are determining our interpretation, which are having top 10 genes based upon these values. We are considering majorly the degree function. So, more the degree, more the importance of the role of that gene in the disease. For example, it was connected to n number of edges, so it has major number of target interventions are present in this disease. So from these 86 genes, we have shown the top 10 targets like vascular endothelial growth factor, EGFR, ESR, estrogen receptor 1, PTGS2. So these are the different targets of top 10 targets which having higher number of degree between as closeness. Based upon that, we have collected the top 10 uh, genes. Just based upon the same protein drug interaction, we have collected the top 10 constituents. So this is the entire network. So you can see in the middle Persia americana, around that you have different constituents, and around that you have the genes. It is an integrated network of protein and protein drug interaction networking. So from this we have identified the from the cytoscape protein interaction we have identified the top 10 targets, and protein drug interactions we have identified the top 10 compounds. Again we have seen what are these 86 common genes we have seen. For that 86 common genes, what are the constituents present? As constituents only we have taken going for the further studies. So based upon this interaction, we have isolated top 10 compounds and top 10 genes. So final step is it is molecular docking studies. So from the beginning, target prediction, we have identified the targets. Second thing, we are coming to the best targets and best constituents. So after that, we are going for the molecular modeling tools, which is molecular docking studies, which is structure-based drug design. We have done in the Pyrex software, and we have seen on two interactions. This is the final step of our work. Like here, what we have seen, we have identified top 10 effective ligands and top 10 genes. So first, already we know the molecular docking process. There are normally four steps involved in the molecular docking, like ligand preparation, protein preparation, identifying of active amino acid residues, and docking analysis. Like how we prepare the ligand preparation, we use the Marvin Sketch software. Here we use this software to prepare the ligands. And protein preparation. First, we have taken the proteins from the protein data bank, and we have prepared by using Swiss PDB viewer software, and identification of active amino acid as per the literature, and we have identified by protein ligand interaction profiler. From this website, we have identified the active amino acid residues. And finally, the docking analysis have been carried out by using the software files. So we have showing now top five targets based upon the constituents. So these are the top five constituents like PTGS2, which is the prostaglandin endoperoxidase synthase to protein, vascular endothelial growth factor A, and estrogen receptor 1, and SRC, and epidermal growth factor proteins. These are the top five targets which we have seen. So from the results, what we have observed, so based upon the constituents what we have collected, luteolin is a drug which is a phytoconstituent which is having the highest score in maximum number of targets. So from this prostaglandin endoperoxidase 2, so having minus 9.3 binding affinity compared to all other targets. So from this we can say that luteolin is the phytoconstituent from the avocado has effect on prostaglandin synthetase 2, which is one of the common target for avocado and rheumatoid arthritis. So maybe this target is good uh, for rheumatoid arthritis reduction. So based upon this, we have identified, and we also compared with the standard. Right now for rheumatoid arthritis, methotrexate is useful, and we also use for the co-crystal for the standard analysis. From this, luteolin shows the best binding affinity. So these are some of the 2D interactions what we have taken. Some hydrogen bond interactions compared to this thing. Luteolin also having good interactions. So finally, what we have developed the new hypothesis. Based upon these findings, prostaglandin endoperoxidase 2 is identified as potential therapeutic target for rheumatoid arthritis, and luteolin from Persea americana is identified as a possible fit. So, what is this prostaglandin endoperoxidase 2? So, from the literature search, from various pathways, we have identified in the normal body, we know prostaglandin synthesis, how it will be happening. So, from the phospholipids and the simple lipid molecules in the cell membrane, which by the presence of phospholipase, it's converted into arachidonic acid, and it have going converting into lipooxygenase, leukotrienes, and prostaglandin synthesis. Here, the prostaglandin endoperoxidase 2, which is helpful for the inflammation, majorly for the inflammation prospect. This prostaglandin is useful. So now what we have identified, this specific target is important for the inflammation in rheumatoid arthritis disease. So for this, which drug molecule we have selected from the avocado, luteolin is a phytoconstituent, which is a useful for this prostaglandin endoperoxidase 2 inhibitory activity. So this is the common target for the both Persea americana and rheumatoid arthritis. PTGS2 was found to be play a prominent role in pathogenesis of complications associated with rheumatoid arthritis. Luteolin inhibits the prostaglandin 2 synthesis gene could find the lower the inflammation of the joints. This is our developed newly hypothesis. So many people can say prostaglandin endoperoxidase is an inflammatory marker. It may help for the rheumatoid arthritis. Luteolin also having some inflammatory properties. What is the new thing? But there is no literature that luteolin is perfectly binding to the prostaglandin endoperoxidase synthase 2 to lower the inflammation. So this is the network pharmacology we can approach to identify which target suits for the inflammation from the which constituents. So from the Persea americana, luteolin is the constituent helpful to reduce this inflammation via prostaglandin endoperoxidase synthetase to target. So this is the newly developed hypothesis. Finally, concluding what we are concluding, the study focusing on identifying potential bioactive compounds in Persea americana for treating rheumatoid arthritis. The study results to identify several bioactive compounds, including luteolin, alpha-tadinol, timurolol, and others, as potential candidates for the treatment of rheumatoid arthritis. Luteolin is present in both fruit and leaf parts of Persea americana. So additionally, the VTGS2 gene was identified common gene between Persea americana and rheumatoid arthritis. PTGS2 was found to play a prominent role in the pathogenesis of complications associated with rheumatoid arthritis and also osteoporosis and cardiovascular diseases. Therefore, blocking this PTGS2 gene could potentially lower inflammation and reverse the harmful effects of rheumatoid arthritis. So, this is the project work what we have concluded. So, this is the structure of prostaglandin endoperoxidase 2 synthetase, and this is the luteolin constituents. So from this work, what we can do, future perspectives, we can employ network pharmacology to explore more. So after this network pharmacology, we have identified the target, we have identified the constituent. So further, what we can do, we can conduct the molecular dynamic simulations to assess the protein stability and investigating novel analogs derived from luteolin, study their SAR and evaluate their activity or develop new formulations for pharmaceutical application using luteolin and other plant constituents, or analyze and quantify avocado extract for cognition and analysis purpose, and evaluate the in vitro and in vivo activities in biotechnology and pharmacology using network pharmacology techniques. So from this, each department have their own activity. If you are a chemist, you can derive new luteolin derivatives and you can go for SAR studies and evaluate activities. And if you are a physics background, you want to develop a new formulation. So luteolin you can take and you can prepare a new formulation with other constituents and go with the activity. And if you are a cognitive, you can extract, you can take the avocado leaf as well as fruit. Both you can extract and quantify how much percent of luteolin is present. And that luteolin is helpful in the in vitro and in vivo techniques. So in vitro and in vivo also, you can evaluate whether this luteolin having this activity or not. So these are some of the future perspectives of network pharmacology. So based upon this work, we have published same as case study what I have explained, exploring potential bioactive compounds of Pericia americana for treatment of rheumatoid arthritis through network pharmacology. Till now, what I have said in the case study, that entire case study we have published in current rheumatology reviews. So we have done the network pharmacology and molecular docking, and we have published this work in this journal. And we also published networking pharmacology in various journals. Like here we in computers in biology and medicine, we have done drug repurposing. We have created the protein drug interaction networking. From this, we have published in computers in biology and medicine. And another publication, drug repurposing for thioredoxin interacting protein, which is a diabetic target. So here we have taken the thioredoxin interacting protein as a drug of choice. Here we done drug repurposing by using taking FDA approved drugs, and we have done drug repurposing study. And one more we have done for ERBB1 and ERBB2 for cancer activity. Also here also we have done the protein drug interaction networking study. And vitamin D analog calcitriol for best cancer therapy and integrated drug discovery approach. Here also we have utilized this network pharmacology approaches. Based upon this, we have published these papers, and these are some of the references what we have taken. And finally, thank you. And before ending to thank you, I want to thank my mentor, Dr. Jupina, for what I have today because of her. And thank you for giving this opportunity for the School of Pharmaceutical Sciences and the University. And finally, if you have any queries, with ending with a small note, like information is not knowledge. The only source of knowledge is experience. You need experience to gain wisdom, by Albert Einstein. Thank you once again for giving this opportunity.
Dear participants, the floor is open for discussion. If you have any doubts, any queries, Sir is available with us. If you want, he can demonstrate things live for you.
Hello. Hello.
Yes, sir.
Am I audible, sir?
Yes, sir. Audible.
Actually, I just want to know that. Actually, I just want to know if we have some drug with us, and
Okay, sir.
we don't know about this drug is acting to other proteins or not.
Okay, sir.
Can we make a protein drug interaction network for that?
You have multiple targets, sir, or multiple drugs.
No, multiple drugs I am having. I just want to search the new target, or I just want to repurpose that drug to other targets. Is it
Yes, sir.
possible through network pharmacology or protein interactions?
Yes, sir. It is possible through protein drug interaction networking, sir. So already you have one target. So whatever the drugs, multiple drugs you have, for
Yeah.
example, it may be FDA approved drugs or plant constituents, whatever it may be, you can
Yes, yes,
see,
yes, yes.
yeah, you can see directly whether it is interacting to the target or not by protein drug interaction networking. So as we shown like protein interaction, like that protein drug interaction also we can see by taking one target, multiple drugs.
Okay. Okay. No, but I just want to explore the different target for the available marketed drug. Suppose I have 100 marketed drug of cancers. I
Okay,
just
sir.
want to repurpose their drug for the different targets. Is it possible through network pharmacology? That is my question actually.
Yes, sir. It is possible, sir. First, you want to take what are the different targets? Is there no, sir? You can take the targets first. What are the targets in the cancer?
Okay.
List out the targets, and first you make the protein
But
interaction test.
target I have to search, sir. These target
You
first
can,
I have to
you
search in
can,
Swiss prediction or some
yeah.
I
This
Swiss
request all,
prediction database.
I request participants to ask the question and mute yourself so that the resource person can explain. Once he explains, you
Okay.
can unmute and ask the question. Okay. Yeah. Thank
Okay.
you.
Thank you, sir. Thank you. Okay. Yes, sir. Please.
Yes, sir. Sir, first of all, you can collect the cancer targets by various databases. Like DigiNet is one of the database. You can collect the cancer targets or which type of cancer you can specify, like breast cancer, prostate cancer, which type of cancer you want to specify. You can specify and you can collect the targets from the various database. One is GeneCards database is there. One is DigiNet databases is there. In this database, you can collect all the targets. So right now I'm showing the Excel sheet. Here you can see the all the rheumatoid arthritis targets. Like that you can collect all the cancer targets from the gene cards or DigiNet. Right now DigiNet web server is not working. That's why I can't demonstrate that one. So you can go after some time and you can search directly. You can go to DigiNet and which type of cancer you want. You enter the cancer. You can collect all the targets. So from that you can identify the targets, then you create the protein-protein interaction. So once you created the protein-protein interaction network, you will identify the top targets. So for the top targets, for example, if you want to go for 5 targets, you take the top 5 targets and go for drug repurposing. Like whatever the you said 100 of marketed drugs, you take that all the drugs and you create a network with each protein for protein drug interaction network. From that, you will get the lead compounds and you can go for the molecular docking and evaluation of in vitro and in vivo studies.
Yes, yes, sir. Thank you. Actually, so first we have to search the DigiNet and gene card for the cancer targets we have to collect. Then we
Yes, sir.
go for the our marketed drug target prediction also. We have to merge both the data. Is it like
Yes,
that?
sir. You can
Okay.
do like that also directly. You can convert into protein drug interaction network also. Yes, sir. What you said is exactly correct. You can go for the target prediction from that drugs also. Then what are the common targets are there? From that also you can go for the studies. Otherwise, directly already FDA approved drugs are there. Each target you take, that all drugs you can do the protein drug interaction network. One target, all the drugs. So which drugs are having more interaction, that drugs you can go for the next studies. Like that also you can go, sir.
One more thing, sir, that when we select the best protein from the protein network,
Yes, sir.
protein interaction diagram, once we select, then my question is that protein we selected, on what basis we are selecting the phytochemicals or marketed drug, and these are the possible marketed drug they will bind to these best hit of the proteins. So what is
Yes,
the
sir.
criteria to select the best chemical actually for our marketed drug in that case? Because protein we identified through the protein interaction network by the scoring all those things, but what about the marketed drug which we have to found to the other target? So what is the criteria to select those particular drug for the new target?
Yes, sir. As you said exactly, once you created the protein-protein interaction, based upon the degree betweenness closeness, we are selecting the top genes. In the same manner, take one target and do the protein-drug interaction network. Same, these are the phytoconstituents which I am showing in the screen. Based upon again degree betweenness closeness, you can identify the top constituents, and you can go for the molecular docking analysis also for further evaluation studies. From that, you can identify the top lead molecules. In that way, you can select the constituents or drug molecules.
Thank you, sir, for nice explanation. Very much.
Thank you, sir.
Excuse
Any
me,
other
sir.
participant?
Yes, sir. I have a question to ask. Hello, am I audible?
Yeah, yes, madam, you can ask.
Sir,
Yes,
I
ma'am.
have
Go
done
ahead.
the
Go ahead. Go ahead.
yes. Sir, I have done complete network pharmacology, and then at the end I have got top 10 genes. So now my
Okay.
question is that how to find proteins for that top 3 genes?
For example, like this, you have collected this vascular top 10 genes. You can see in the screen. So like this also, you have collected the top 10 targets, right?
Yes, no, top 10 genes. At the
Yeah,
end of
top 10
network
genes.
pharmacology, we got genes, na sir?
Yes, madam, genes, nothing but targets. Also, we can call them targets also. So these are the genes, what we have seen, that vascular endothelial growth factor. So these are the genes. So how to find this target? Like, for example, you need to select these genes and paste in the UniProt database. So from that, you can identify the target. I will show one example for your case. So for example, this is the gene. So go to UniProt database. So here is the UniProt database. So here you paste that gene, whatever you getting top 10 genes from, that gene you can paste in this search. So here you can go for the humans, as here vascular endothelial growth factor A. So you need to see. So for example, here vascular endothelial growth factor C. Yeah, second one is, which is A, vascular endothelial growth factor A. This is the gene. So if you go inside about this information, vascular endothelial growth factor, you will have everything like function, name, taxonomy, cellular location, what are the disease. And here, if you want to identify the protein, like PDB database, exactly want to that one only, man. PDB, what is the target? So from this, you can identify this vascular endothelial growth factor and their target proteins. These are the target proteins. Here you can identify n number of targets you can identify through this. So these are the separate PDBs. Which PDB, if you want, you can select that one based upon the certain criteria. Hello, is it okay, ma'am?
Sir, for moving for docking, we have to need protein. हो ना सर?
Yeah,
प्रोटीन लागतील ना आपल्याला त्यासाठी?
we need a protein, madam. We need a protein.
आणि मग युनिप्रोट आयडी वरून आपण प्रोटीन बघू का जीन्सचा?
Ma'am,
Can
you can
you
ask
repeat?
question in English, please.
Sir, at the end of at the end of network pharmacology, I got top 3 genes. According to your opinion, I have to search UniProt ID of that top 3 genes, and then I will move towards the docking, right?
Yes, madam. Exactly.
But my
Will you-
yes, but my question is for docking, we require proteins.
Yeah, madam.
So is it helpful to get protein through that UniProt ID?
Yeah, madam. Here you can see my screen is visible now, madam. Here structure is there now. You can click the structure
Yes,
here.
sir.
So this is the gene complete, one of the protein of vascular endothelial growth factor. Down here, many PDB IDs are there. 1BJI, which is one PDB, 1CZ8. These are separate PDBs. Based upon your criteria, you can select the PDB for the molecular docking studies.
On the basis of which criteria we have to select one of them, one of the
Yeah.
protein?
How we need to select the best PDB. So there is certain criteria, like you can go to the protein data bank. For example, 1BJ1 is there. It should be X-ray diffraction method. There are different methods. So one of the method is X-ray crystallography method, and the resolution you need to see here. The angstroms is the resolution. It should be majorly below 2 or 2.5. If below 2.5, the resolution of the protein is there, means it is a good PDB protein. And it should be, for example, it is a Homo sapiens. Homo sapiens mutation should not be present. So based upon that criteria, you can select the PDB.
Okay, sir. Thank you, sir.
Yeah, thank you.
Sir, I have one doubt regarding you selected the panel targets which are common between the compound and the disease, right?
Yes,
So
sir.
now,
Yes, sir.
now I have only one disease. Like I'm working on a non-alcoholic steatohepatitis. So from that, how can I select the targets? Means how to carry forward from that?
Once again, can you say, sir? You have the disease hepatitis.
Uh, yeah, non-alcoholic steatohepatitis. I have one disease. So I don't
Okay,
have any
sir.
compounds. I'm just looking for which targets which I can target. So how can I carry forward that?
Already you said you have collected some targets, right?
Yeah, some targets I have collected. So
From where
based
you
on that
have collected,
three,
sir?
sorry,
From where you have collected the targets, sir?
sir, I have used the three databases like Gene Cards, CTD, and DSGNet. And from
Okay,
that, and to validate further, I have used bioinformatics one also database. But now,
okay.
how can I carry forward to select the prominent target from that?
Yes, sir. Already you have collected the targets. For example, 500 targets you have collected. For example, I'm saying from the various databases you have collected for the hepatitis,
Yeah, yeah, yeah.
500 targets, for example, you can see. First you create the protein-protein interaction network. So from
Okay,
there
sir.
you can identify the top targets based upon this degree between this closeness in the cytoscape software. Directly you can import that all the 500 targets in the cytoscape, and you interact with protein-protein interaction network. Based upon that degree between the closeness, you can identify the top targets. And once again, the targets you can go from the different pathways like reactome or keg pathway or literature search. Already the target having some literature, you can go that literature. Okay,
Okay,
this is the good target. Like that, you can select the best target. Or if you want to
okay, sir.
validate further more, this target you take to the molecular docking studies also. There you can see which target having more binding affinity for your particular compounds or particular phytoconstituents.
Okay, sir. Yeah.
Sir, I have one doubt, sir.
Yes,
Sir, like as I am going for network pharmacology approach, so I got top 10 targets. Generally select based on betweenness, because if I am going on degree, top 10 targets are getting changed. Because
okay.
so what I should do in that case? I should go further with the betweenness top 10 targets or with a degree top 10 targets.
So actually, majorly when we are conducting the next studies, like after network pharmacology, we are going to the molecular docking studies. Actually, this doubt we also got, we have raised it between sometimes degree, sometimes. So we have carried forward top targets for betweenness, top targets for degree, and we go for the molecular docking studies. There we find which are getting the good degree scores, they are getting good scores in molecular docking. So from the literature also, they are saying degree is the best score for the uh, target selection. And in the molecular docking studies also, we find the degree score is better for going through the next step. So you can take degree as the best choice of interest, and you can go forward.
And sir, like in my top 10 targets, two targets, although I am getting the common between disease and phytoconstituents, but when I went for literature search, those two targets are not involved directly in the progression of the disease. So after I get top 10 targets, should I continue with those two targets, or when I am going for docking, I should eliminate those two targets?
Slowly, sir. Once again, slowly you can say, sir.
So, like, for example, in my top 10 targets, CA I got as a gene, like my target, but that
Okay.
target that is not involved in the progression of the disease. So, when I am going for molecular docking, should I continue with that or should I eliminate it?
Whenever you collected the target from the disease, automatically it will involve in some of the progression, the target. You
It
can
is not
find by
directly involving. It's like after two
indirectly
or
maybe happens.
yeah.
Yeah.
So should I continue with that or should I eliminate?
For example, if that having no literature search, no activity, if you do, it will be the best research according to my knowledge.
Yeah.
So if there is no activity till now for that disease for through this target,
Yes,
so
sir.
it will be the novel hypothesis for developing the new research hypothesis. So
Yes,
it
sir.
will be helpful if you conduct to these targets also.
Okay, sir. And sir, one more doubt, sir. As we go for Lipinski rule of five for
Yes,
selection of our phytoconstituents, like those who are following, we will continue with that. But
yes, sir.
beside that, on literature search, I found that some more phytoconstituents which are present in the plant which I have selected, but they are not following Lipinski rule of five. So after can further can I include those phytoconstituents also, or should I totally eliminate those?
No, sir. Actually, what now? Out of five, you
Yes,
can
sir.
go till two violations is not a problem. Out
Yes,
of
sir.
five, till two violations is not a problem. You can go further studies.
So which two violations should I consider, sir, from?
Any two, sir. Any two. Like out
Yes, sir.
of five violation, you can see the two. Because
Yes,
if
sir.
you are going for the formulation, you
Yeah.
want to see the which type of involve like partition coefficient, whether it is a lipophilic drug or hydrophilic drug. Based
Yes, sir.
upon that criteria, you can eliminate which two violations you required.
Oh. And sir, should I consider the molecular weight also in mind when I am selecting the phytoconstituents if they are not following Lipinski rule? Because our route of administration also is a thing in which molecular weight. So should I keep that in mind or should I eliminate that thing? And because as I am going for further formulation development, so that's what I'm.
See, sir, if molecular weight is increasing, there is a bioavailability problem will be occurs.
Yeah.
So if you follow the molecular weight and partition coefficient, it will be good for the formulation aspects.
Okay.
So you
Thank you,
can
sir.
follow this Lipinski rule of five. It will be better.
Thank you, sir.
Thank you.
Thank you, sir. Thank
Sir,
you, sir.
there are some questions in the chat box. You can address them.
Sure. One question from Gayatri, sir, can it be used for newly designed molecules for multiple targets? Newly designed compounds, I will not suggest right now. Already FDA approved drugs is there means you can go for the multiple target protein-protein interaction because newly designed molecules, it is not coming good results when we have seen, but it should be. Right now, I don't have that much knowledge about newly designed molecules. What we have tested, it is not good. So already FDA approved drugs like whatever available in the market, you can go for that multiple target. So already this I have explained. It is possible to apply network pharmacology for finding best prebiotics in treatment of any disease. Finding best probiotics in the treatment of any disease. Yes, you can go what probiotic, what are the different constituents are there in the probiotic, what are the things you are taking in the probiotics. Based upon that, you can create a network and you can go for the disease. That is also one good way for this one. Sir, in GO, like genome analysis, how we will get the p-value in enrichment analysis? When you go for the enrichment analysis, the statistical values from the previous literature, you will get the p-value, statistical values completely from the previous, what are the data in vitro or in vivo data we have there. Based upon the p-value, you can identify. Next, sir, can you show me how to get protein of AKT1 gene? As already I have shown one of the gene like vascular endothelial growth factor in UniProt. You can go to the UniProt database. In UniProt database, you can select that AKT1 gene. You will get the protein. If you want to the PDB, you select the structure option. So from that, you can select the PDBs. If we are going for any disease study, it is necessary to find protein as per disease, or we can select any protein. we can't select any protein. If it is related to the disease protein means it will be very fine. So you can go for the disease related proteins. So you'll get the uh, best output of the research work. So that's all, sir, in chat box, the questions.
Any other questions from the participants? Any other questions?
How
Sir, I
we
have a
find?
personal question to ask.
Okay, sir.
Let us assume that a M Pharma student, a B Pharma student who has no information about network pharmacology, he want
Okay,
to go or pursue his research in network pharmacology. He has no idea about which disease he has to select, which plant he has to select. So where he or she need to start with?
yes, sir. Very good question, because as a beginners of the to the research, whether a B Pharma or M Pharma, every faculty will think, well, you can select one topic, based upon that we can go forward. So this network pharmacology will help them to identify from the scratch. So for example, any B Pharm project, if you want to take, they will say directly diabetes, or rheumatoid arthritis. So like different diseases names they will say. But first, based upon that, from the literature survey, we need to start anything. Once we need to select any disease or any plant constituent or anything what we are to select, first starting thing is literature search. So if you want to select any diabetic, why we need to select the diabetic? So till now, what are the drugs that are available? What is the research gap in that diabetes? What is the till now marketed available drugs from the phytoconstituents? Which phytoconstituents are helpful for the diabetes? So there should be some research gap. So till now, what are the targets they have identified? Which targets having more value for the diabetes mellitus? Or like that, if he knows some literature search, so he can get one idea regarding, okay, this area having some research gap. Based upon this, we need to select this disease. So till now, there is no much information in the literature. So if you take this project, it will go further studies. So for them, the network pharmacology will be helpful, because this network pharmacology, you can identify the vast number of targets. He can expose many number of signaling pathways at the beginning stage only. So while going for the in vitro and in vivo studies, he will not confuse any problem. Already he has clarity which target he is focusing in which disease. So directly he will go for that signaling pathway, that activity he can conduct very nicely. In related to the selection of a drug molecule or any phytoconstituent, the same pattern. As the beginning, we need to go for the literature search. So once he want to do for the formulation, directly which drug he want to take, like he can go with FDA approved drugs. What are the available FDA approved drugs? He can do drug repurposing study, like protein drug interaction study. So he can collect all the drug molecules. He can incorporate into the what are the targets he identified from this, and he can go forward. So for the beginners, it is a very good start to them for the in silico molecular tools. From my side, it is the best choice of network pharmacology to start their career.
Sir, we have one more question in the chat box. If network pharmacology is applicable for probiotic screening, same some software applicable whatever you have applied for traditional drug screening and target identification.
Okay, first of all, which type of probiotics? Actually, we didn't went completely to the probiotics. Directly, we are going for traditional medicines. So we need to search what are the databases are helpful for the probiotics. First, we need to search from the literature only. Right now, I also don't know which databases are available for the probiotics. So if they are taking any constituents in the probiotics, the impact database is helpful for that, and they can start the collecting of the target constituents from the database. If any specifically database is there, I want to search. Right now, I don't know what is exactly what prebiotic databases are there.
Good morning, sir.
Good morning, sir.
Sir, my question is something different from the network pharmacology. If a formulation we can give to any patient, but formulation containing so much of excipients and other stabilizing agent and compounds, so they can interact with the protein, and there is also a study for this type of excipients also, sir.
Excipient studies, it is a very good question, sir. Like excipient interacting with the protein. It is also a very good question. Right now, exactly the excipient interacting with protein studies we can do or not, I didn't try it. So we need to see any some of the literature previously have been conducted like this. It is a very valid question, and we need to try, sir. So if you have time, you can try that one and go forward, sir.
Thank you, sir. I am also researcher, sir. So thank you, sir, for the giving the insight, sir. Thank you.
Thank you, sir.
Sir, one more question. Do free software's results are accepted for good impact factor journals?
Yes, who asked this question? Very good question. Everyone will have the same query. Free softwares having the things accepting range. So now I'm showing the paper publication what we have done. So all the publications what we have done from the free softwares only. Only for molecular dynamic simulations we have used the Schrodinger. So remaining all the softwares are free. So various web servers, various softwares what we have used is completely free softwares. So in this paper, we have used a completely cytoscape, which is free, and web servers like Impact, what I showed today. So this paper is completely comprised of free softwares only, nothing paid softwares. We have used all online web servers and free softwares. So here we used only the Schrodinger for the dynamic studies. Remaining rest all event docking also we have conducted in the Pyrex, and we have published this paper with a good impact of 7.7 impact factor. And this is drug repurposing studies also, same like molecular networking, pharmacophore modeling, and molecular tracking approaches. All these three approaches also we have done in the free softwares. Molecular networking via Cytoscape, pharmacophore modeling via PharmaGist software, and zinc pharma virtual screener, and molecular docking by using Autodoc or Pyrex. We have used this paper. So for this paper, repurposing of FDA approved drugs, same Cytoscape and Pyrex we have used for this paper also. So vitamin D and log. Here also we used some other softwares like free softwares only. For molecular dynamics only, we have used the Schrodinger software. Rest all we have used the free softwares only. If you have GROMAX, if you have applicable, you can go for the molecular dynamic simulation in GROMAX, which is free. So we can publish lots many papers with good impact factor papers.
Any other questions? Any other questions? I think all your questions are cleared. Thank you, Swarup sir, for your.
Hello, sir.
Yes, ma'am. Proceed.
Sir, I have a question.
Yes, madam.
Sir, can you please tell me how to find disease-related protein using Uniprot?
Disease
Disease-related protein using Uniprot.
Uniprot. I will show once again that how to take that. So you have seen my presentation slides, right? So these are the top what we have collected, the top 10 targets. So from this you want to identify the proteins. So same, for example, I am showing this vascular endothelial growth factor. So just copy that gene and go to the UniProt database. So already this is the UniProt database. I am closing and opening again once again. So you can go for the Google and you click the UniProt. So once entering into the UniProt, here you can search that, for example, this is a vascular endothelial growth factor A. You can go for the search. Here you can click, many options are there in the left side. You can go for the humans, means humans. If you are going with another means, you can select the other things. So here there are many things like vascular endothelial growth factor C, A, D, but we need vascular endothelial growth factor A. So we need to select this one, vascular endothelial growth factor. This is the gene name, vascular endothelial growth factor A. This is the gene which you need to select. So for this, you need to have the target proteins. What are the target proteins are there for this vascular endothelial growth factor A? So once go inside the thing, you can have the brief thing about vascular endothelial growth factor and what is the function. Here in the left hand side, you can see the structure. One name which is structure. You can click the structure. Here you can identify the protein of vascular endothelial growth factor protein. Here for molecular docking, if you want to do, you want to select the PDBs. Here are the some PDBs you can select via resolution, mutations, X-ray, experimental method, or previous literature. What are the PDBs they have used? You can select any one of the PDB and you can go for the molecular docking. Is it clear, madam?
Yes, sir. Thank you.
Yeah, thank
Yes, Swaroop sir, on personal note, I would like to ask one more question. You have discussed some 9 to 10 different softwares, right? Starting to ending, right?
you. Yes, sir.
Now, all these softwares, for learning all these softwares, do we need to have any technical knowledge? That is my first question. And the second question is, how much time would it take for a B Pharma student to learn those softwares? Number two. Number three, if he learn all those softwares, what he can do as far as network pharmacology is concerned? Kindly address this, sir.
Yes, sir. It is a very great question. So many people want to learn the softwares. So before learning the software, why we need to learn the software? He should know the B Pharma student or M Pharma student. He should know in what aspects. If, for example, I am showing in the screen, there are many web servers and softwares like Impact, Open Bible, Data Warrior, Binding DB, DCNet, Venue. These are the several web servers and softwares, right? So whether starting of the B Pharma person or M Pharma person, why? What is the need of the software? First, he need to know. Technical knowledge means first you need to know what is the output of the software. So for example, if you use this one, what you will get output. So minimum basic knowledge he should have before utilizing this one. Once he knows, he can start the using. And one more thing, how much time it required to learn these softwares. It is completely depend on the person. I can't say this is this many days, this is this many days. So it's completely depend on the person. So if he was interested more, he can explore the software within a week, within a 3 days, or within a month. So if you want to go for each web server, he need to practice more times. So for example, Impact database is there. He need to go to the Impact database. He need to search the various plans. You need to keep various plans. What are the constituents are coming? You need to practice number of times. If you practice means it will be easy for identification. And various software like Open Bible, he need to explore, sir. Like he need to practice 1 or 2 times. So many YouTube videos are there how these softwares will work. So he can directly go to the YouTube videos how it is opening the opening Bible. So every student will come to know when he needs the work. Then only it will be possible. Like I required to convert SDF to mole format. Then I should know how to convert this molecule, what software is available for conversion of this molecule. Then I will go for the search. So there is the open Babel software. Then I want to explore. So it will take almost 3 months of time for this network pharmacology. Completely he want to learn this from the starting to ending of interpretation of the data, selection of the compounds, software usage. You need to explore the more options inside. For example, what we select means which option is selecting. So what is this output? Entirely our research is output. So each and every option, if you know, it will be better for the student, according to my knowledge. Because when I start preparing or learning, each and every time I have utilized a number of times. So Open Bible means I have utilized 100 compounds. I will take in different formats I have downloaded, and I will convert into different formats. So I will be proficient in that software. Like Data Warrior is there. So many compounds. How so previously I used Swiss ADME for ADMET analysis. So in that suicidality, what I have seen the disadvantage is each molecule I need to incorporate or multiple molecules we can incorporate and we can see data. Each data I want to correlate piecewise. In data warrior, what I have seen easier. So all multiple compounds, if you are incorporating in the single time, we can filter all the molecules. So by using the different softwares only, we can know this will be easier way to go for faster the research like that. So if we want to learn, it will take 3 months of time for complete network pharmacology in the each and every aspect of the things.
Thank you, sir. Thank you so much for your answer.
Yes, sir.
Any other questions? Last call. Any other questions from the participant side? I think no more questions were left. Thank you, Swarup sir, for your database for TB. Database for TB. Vijay, what do you like to ask?
Is there any sorry to interrupt.
Yeah, you can. Yes.
That can give us targets of TB. For example, well, we are getting for example, we are using superbred, right? And it is giving targets of humans. So just like that, is it possible that we get targets of TB that can be targeted by our compound based on our
TB means tuberculosis. You are speaking about tuberculosis, right? Hello.
Yes, sir. Am I
Yeah.
audible?
Yes, sir. Yes, sir. You can get the targets from the disease-oriented genes. You can get the targets from this genet. You can get targets. All the targets you can get.
So
Otherwise, you
based
can
on
go to the Otherwise, you
no,
can go to the Finch database. There, if you select the instead of Homo sapiens, if you select any microorganism, you can get that targets from the strings.
in which
String,
string?
string, sir. String.
So string, I tried string. I tried string, but they are not showing any targets for TB. I tried that. I know we can change that Homo sapiens to other multiple organisms, but they are just
Okay.
showing only one target. That is DHRF. That is dihydrofolate reductase enzyme. Right.
Okay.
So is there any other database which can give me the targets
You want to
just
try
like
with
this?
DisGenet also. You can try DisGenet one and GeneCards one. You can try with that websites also. You can find
What
some
I'm
more things.
saying is, yeah, I got your point. But what I'm saying, this will give me all the proteins which are present in TB, right? Which we can target.
Yeah,
The
yeah, yeah. You
question is,
can get.
we are using superbred, right? For example, we are using superbred, and superbred will give us targets based on the structure of our compound, right?
Okay.
तो आई एम जस्ट आस्किंग, इज इट पॉसिबल कि हमारा जो स्ट्रक्चर है कंपाउंड का उसको उसके बेसिस पे हमको टीबी के टारगेट मिले? ये जो हमको सुपर प्रेड से टारगेट मिल रहे हैं या बाइंडिंग डीबी से मिल रहे हैं, वो सारे ह्यूमन के टारगेट हैं। तो सेम इज इट पॉसिबल कि हमको टीबी के टारगेट्स मिले, क्योंकि टीबी के प्रोटीन्स ये टारगेट हो सकते हैं बाय आवर कंपाउंड? Is there any database? I asked this question previously also, and sir also told me one website, but that is also not working. So
Sir,
if you know any,
particular compound, particular disease, you will not get separate targets, sir. By using these techniques only, you can identify a new targets directly for one. For example, methotrexate, rheumatoid arthritis. For this particular target, if you want to identify this, it will be a challenging process. You can conduct this networking and all the things, and you can find that target, which target is important. You can identify the what are the targets for methotrexate, and we can identify the targets of TB, and which are the common target. Based upon that, you can find the final ultimate target. Directly, there is no website to identify the drug and disease target. If the drug and disease target directly, we can take that target and we can go for the studies. But till now, according to my knowledge, there is no that type of website directly for the disease and drug.
okay. So sir, we use superpred, right? We use the superpred. What the superpred is doing, when we upload our smile structure in superpred or binding DB or protonics, right? They all are
Yeah.
giving us probable target.
Yes, sir.
This all probable targets are for Homo sapiens. Sorry, this all probable targets are for Homo sapiens. I am saying,
Okay.
is it possible that I get targets for Mycobacterium tuberculosis species? I did not find any database which is providing targets of Mycobacterium tuberculosis. So, is there any database? There are few databases, but they are not working currently. I don't know why, but they are not working currently. So, if you are aware of any database which can give me targets of Mycobacterium tuberculosis series.
Sir, instead of selecting that various databases, whatever the target, according to my knowledge, I am saying, whatever the targets you are getting, identify that protein in the PDB. In the PDB, you will get different options like whether it is an Homo sapien bacteria. From there, you can scrutinize whether it is Homo sapien target, Mycobacterium target.
Yeah.
Like that, you can segregate there.
Got it, sir. Got it. Got your point. Should do that. I'll do that, sir.
Yeah. Thank you, sir.
Yeah.
So 2 more questions in the chat box, if you could answer them.
Uh,
Can we download
we
the screened molecules from Data Warrior at a time?
Data warrior is for the ADMET properties. From that ADMET properties, all we can download at a time the things, but compound structures we can't download at a time. There is for uploading all the compounds at a time. You will get all the information about ADMET properties like molecular weight, partition coefficient, solubility, hydrogen bond donor, etc. Every data it will come within one click. So by using that software, you will get the data. Not molecules you can't download from the data warrior software. You can go for the PubChem, or whether you are going for the impact database, from there you can download the compounds. So for that, I can show you how we did show that.
Please wait, sir. I'll give you the host. Now you can try, sir.
Is it visible for screen?
No, sir, no.
Okay. In data warrior software, you can predict the ADMET properties and you can download the data. You can't download the compounds. Compounds we need to incorporate. Okay. And merge our study. What it means merge for the study means, can we get targets from multiple databases like TTD and DGD? Yeah, you can get multiple targets from DigiNet binding DB. Yeah, you can take the targets from multiple databases. That is not a problem. But DigiNet is used for the disease targets and binding DB is used for the plant targets or drug targets.
Is
For
it
merging,
possible to download? Yes, sir,
is it
proceed.
possible to download?
All softwares freely, whatever you have shown in slide number 12.
Yeah, sir, you can download all the things from the online itself. You can go to Google and you can download it by keeping that name.
Okay. Thank you, sir. I think all the participants have cleared their doubts.
Okay.
Yeah. As a token of appreciation, sir, please accept our.
Yes, sir. Yes.
Is it visible, sir? Please confirm.
Yes, sir. It is visible.
Yeah. Yeah.
Thank you very
Thank
much,
you.
sir, once again for
Yeah.
giving this
Thank you.
opportunity.
Yeah. Thank you, Swarup, sir. Thank you so much for your time. Though you are not in the university, you are in Ahmedabad with official work. But even then, you have taken time for us, addressing our participant, giving very good inputs regarding network pharmacology. Especially, you have driven all the participants through different softwares, how to use the softwares, how to download them. You've shown live demonstration.
Yes.
I believe all the participants have cleared their doubts. They are eagerly waiting to have their hands-on on the softwares what you have showed. Thank you, sir. Thank you so much for your presentation. Thank
Thank
you,
you,
sir.
sir.
Yeah. Thank you, participant. We'll meet tomorrow.